Hello friends, good afternoon. Welcome to CEC live lectures. Dear friends, in this session the topic for discussion is genetic recombination and bacteria. I would like to tell you that this lecture is a part of ongoing lecture series on genetics. To take this topic, we have our subject expert Dr. V.S. Rawat. Dr. Rawat is Assistant Professor, Department of Zoology, Hindu College, University of Delhi. With this, I would like to request Sir to begin his lecture.
Thank you.
Welcome.
Friends, today I will be talking about a very important aspect in the genetic history or genetic life of the bacteria, that is recombination. Recombination is a process which brings about new genetic mix-up or new genetic arrangement of genes in any organism. Now, for eukaryote, this process is very simple, as this process occurs in sex cells, that is ova and sperm, during meiosis, where homologous chromosome pair and crossing over occurs. Whereas in case of bacteria which do not have sexes, this process cannot occur by meiosis. So there are various methods through which this particular genetic recombination process can occur. I have already talked about two such methods, that is conjugation and transformation, which leads to genetic recombination in bacteria. Now I will be talking about the third important topic, that is transduction. How transduction leads to recombination of genes, that is making of new arrangement of genes in prokaryotic organisms. So the outline for today's lecture would be, I've already talked about recombination. I'll be giving a summary of what exactly recombination is, and basically I'll be talking about how the process of transduction was discovered. So discovery of transduction as a process of recombination will be the main highlight of this particular lecture, and then I'll be talking about types of life cycle of bacteriophages. There are two types of life cycles of bacteriophages. I'll be discussing about them, and I'll add a note on significance of bacteriophages in genetic recombination as well as for biotechnological aspect. The learning objectives for this lecture would be to know about the historical perspective of how transduction as a method of recombination was deciphered, and also learning about the life cycle of bacteriophages. So recombination is basically the process by which the pieces of DNA are recombined to give rise to a new combination. And that is why the terminology recombination is used here. In eukaryotic cells, recombination occurs during meiosis when the homologous pairs of maternal and paternal chromosomes align. After this alignment, during meiosis, the crossing over occurs between these chromosomes, which leads to exchange of genetic material between the maternal and paternal chromosomes. Now the offspring which are produced because of this particular after the mating, the offspring which are produced, they have a new combination of genes which is very much different from their parents. So there is a great degree of variation, because first of all there is recombination in the father, in the sex cells of the father, as well as there is recombination in the sex cells of the mother. And when these recombined chromosomes, they come together in the zygote, then a new degree of variation is occurring in the zygote. So the offsprings are very much different genetical arrangement from their parents, be it mother or be it father. Now the recombination is required to create genetic diversity at the levels of genes that reflect differences in the DNA sequence of different organisms. So if there was no recombination, no evolution would have taken place, and there would not have been any formation of new species through variation. If there was no variation, no new organisms, no new species would have evolved from the previously existing species. Now we all know that prokaryotes do not have male and female sex, and they do not reproduce sexually. They undergo asexual reproduction. But there are methods by which recombination does occur in prokaryotes. and it has been characterized in a number of species, and it has been very well characterized in Escherichia coli. The three mechanisms through which conjugation occurs in prokaryotes can be named as conjugation, transformation and transduction. Now all these three methods have different requirements. They proceed in a different manner from each other, and obviously they will be having a different set of requirements. And the success rate of all these methods also varies depending on the conditions which are present in the environment for the bacteria to undergo genetic recombination. Now talking about conjugation, the probability of occurrence of this particular mechanism is quite low, because in this case the donor bacterial cell must contain a conjugative plasmid. The cell which is going to donate its DNA to the recipient cell, it must be having a particular plasmid known as conjugative plasmid or F plasmid. If this plasmid is absent in the donor bacteria, then recombination will not be taking place. Also, there is the necessary condition of establishment of a physical contact between the donor and the recipient. If there is no physical contact between the donor and the recipient, then there won't be any transfer of DNA from the donor to the recipient, and if there is no transfer, then recombination cannot occur, and new arrangement of genes cannot take place in the recipient bacteria. Also, the third requirement is the essentiality of metabolically active cells. If the cells are not active metabolically, then the DNA transfer, even if it takes place, the synthesis of complementary strand cannot take place, and if that doesn't happen, then the recombination doesn't occur. The second method by which recombination occur is transformation. It is another method of gene transfer from the environment to the bacterial species. In this particular case, there is no essentiality of formation of physical contact between the donor and the recipient. The naked DNA which is present in the environment can be uptaken by the recipient cell, and once it is uptaken, then there can be homologous recombination, and that particular piece of DNA which has been uptaken can be inserted into the chromosome of the bacteria. So in this particular case, there is no requirement of forming a physical contact as such. So gene transfer by transformation does not require a living donor cell, because release of DNA during death and cell lysis can provide free DNA, and this free DNA can be uptaken by the recipient cell. However, the persistence and dissemination of DNA in the environment determines how far in time and space the recipient cell can be separated from the donor, because we know that DNA as such is very susceptible to digestion by DNA cells, and it cannot persist for a very long time in the environment. So if a bacterial cell is lysing and releasing its DNA in the environment, in which lot of DNA is present, then that DNA will be cleaved or digested, and no DNA will be left for uptaking by the recipient bacterial cell. So in that particular case, there won't be any transformation. So the essential requirement is that DNA should be kept stable for a long period of time. Third requirement is that the recipient must be physiologically active to be able to uptake the DNA. If the recipient is not physiologically active, if it is dormant or if it is in a quiescent stage, then there cannot be any DNA uptake and there cannot be any recombination. Also, there has to be a close genetic, there is no requirement of a close genetic relationship between the donor and the recipient, because any DNA which is present in the environment can be uptaken by the recipient cell. Now, the third important mechanism by which genetic recombination occurs is transduction. The transfer of genes by transduction requires a metabolically active donor cell in which transducing phage particles are produced during viral reproduction. As the name suggests, as the definition suggests, that transduction essentially requires presence of bacterial viruses known as bacteriophages, and these bacteriophages have to infect the bacterial cell, and after the infection, the DNA material of the bacteria will get packaged into the head of the bacteriophage, and this bacteriophage, when it infects the next bacterial cell, that is the recipient cell, it will release the DNA into that bacterial cell, and from that recombination will take place. However, the recipient can be specially and temporarily separated from the donor, because the genetic information in the transducing particle can persist. In case of transformation, the recipient it is necessary that recipient stays near the environment where DNA is present as such, whereas in case of transduction, it is not necessary that the bacteria be present in the immediate environment, because the DNA is safe in the bacteriophage head and it won't be degraded. However, the bacteriophage has to make a physical contact with the bacteria at some point of time for transferring the DNA which is packaged in its head. Now, the another important aspect is that the recipient must be related to the donor by common sensitivity to the bacteriophage, because if a particular species of bacteria cannot be infected by bacteriophage, then genetic recombination cannot take place. So, it is important that both the donor and the recipient species they are able to be infected by the bacteriophage. That is a very important aspect. But extensive metabolic activity is not required for the infection process. So, transduction is another very important aspect where bacteriophage play a very important role in transferring genetic information, and which leads to genetic recombination. But this particular thing, that is discovery of transduction, did not occur till 1950s. And how did the discovery of transduction occur? Now, I will be talking about how the scientists were able to discover this mechanism of transduction which leads to genetic recombination. Now, with the discovery of penicillin enrichment technique, a number of auxotrophic strains of Salmonella typhimurium were isolated in the 1950s. Auxotrophic strains are those strains which are not able to synthesize some essential or important nutrients, and they require presence of these nutrients in the minimal media. Now, the penicillin enrichment technique was discovered or developed, and with the help of that, a number of auxotrophic strains could be isolated. Similarly, mutant strains, that is auxotrophic mutant strains of Escherichia coli K12 were isolated. This is another strain of E. coli K12. Numerous auxotrophic strains of this particular strain were isolated by similar enrichment technique. In 1948, S. typhimurium strains arrived from the laboratory of Lillingen in Sweden to laboratory of Lederberg. So, this specific type of strain of S. typhimurium from the laboratory of Lillingen in Sweden was sent to laboratory of Lederberg in USA. After that, work was carried out in this particular strain, and a large number of auxotrophic mutants from these strains were obtained by the penicillin enrichment procedure. In October 1950, Zinder, who was a student of Norton Zinder was his name, was a student of Lederberg. He made a cross between two of the Lillingen strains of Salmonella typhimurium. These strains were named as LT2 and LT22. Now, when the cross was made between these two strains, it yielded recombinants at a frequency even higher than previously observed for E. coli crosses, and the frequency for E. coli was about 10 to the power minus 6 to 10 to the power minus 7. Now, what was happening? To understand this, he performed a YouTube experiment. This type of experiment was earlier done by Davis using E. coli K12 strain, where also he was trying to understand why this particular recombination is occurring in E. coli strains. Now, in this particular YouTube experiment, two different cultures were grown in the separate arms of a glass U tube. The tube of glass was shaped in the form of alphabet U, and the two arms of the U had different cultures, different strains of the Salmonella typhimurium growing in them. And between the arms was a sintered glass filter with pore size small enough to prevent bacterial passage. The two arms were separated by a glass filter, and the pore size was very small, so that bacteria could not transfer from one side of the tube to another side. The medium was flushed back and forth between the arms as the bacteria grew, and those in each arm were sampled periodically for existence of prototrophs. Now both LT2 and L22 were auxotrophs, and periodically they were, the arms of the U tube were sampled for presence of prototrophs. The LT2 parent was an auxotroph which required methionine and histidine, while the LT22 parent was a phenylalanine and tryptophan auxotroph. Now it was found that prototrophs were recovered at a frequency of 10 to the power minus 5 from the LT22 side, but they were never recovered from the LT2 side of the tube. Now what exactly was happening? This is the experimental setup diagram. It shows that there is a U tube, and on one side is growing LT22 strain, on the other side is LT2 strain. LT22 is phenylalanine and tryptophan auxotroph, and LT2 is methionine and histidine auxotroph. Medium is passed back and forth through the sintered glass filter, and LT2, from the LT2 side, prototrophs could never be isolated, whereas prototrophs could be isolated from the LT22 side. Now what exactly was happening? If conjugation was responsible for the recombination, then the prevailing experimental condition would prevent formation of recombinants, because as we know, for conjugation to occur, there has to be physical contact between the bacteria, and because of the filter present, the two types of strains, the bacteria on the two sides could never make physical contact. When the two cultures were grown together, LT22 mutants were converted to prototrophs without any effect on the LT2 mutations. It was suggested that some filterable L forms which looked like spheroplast and were induced due to stress were responsible for bringing about this recombination. When many of the Salmonella strains were stressed with chemicals such as crystal violet by aging or by the growth of certain Salmonella phages, varying amounts of activity was produced, which was known as filterable agent. So the agent which was carrying out this particular recombination was termed as filterable agent. The filterable agent was produced by LT2 cells only when they were grown in association with LT22 cells. If LT2 cells were grown independently and the culture medium was added to the culture of LT22 cells, no recombinants were ever recovered. So LT22 cells played a role in production of filterable agent by LT2, and they do so only when grown in a common medium. Presence of DNAs did not render the FA inactive. This suggested that recombination was not brought about by transformation, because for transformation to occur, naked DNA has to be present, and if DNA is added to the medium, if naked DNA will be present, then it would be digested by DNA. So both conjugation and transformation are ruled out from this type of genetic recombination. The FA could not pass through the filter of U tube if the pore size was reduced below the size of a bacteriophage, that is 0.1 micrometer. This suggested that bacteriophage is the medium through which this particular genetic recombination event is taking place. So what exactly are bacteriophages? Bacteriophages are those viruses which infect bacteria. Now these bacteriophages have a typical structure. They have a head which is made up of protein, and this head packages DNA, and then there is a neck which joins to a tail through a collar, and this tail is covered by sheath. Most of the bacteriophages, they will be having tail fibers which help in attachment of these bacteriophages to the bacterial cell, but there are bacteriophages which do not have tail fibers. So this is the basic structure of the bacteriophage. Now the first step in the infection process in the life cycle of bacteria is the adsorption of the phage to the bacterial cell, which is mediated by tail fibers or by some other analogous structures on those phages which lack tail fibers. Now phages attach to specific receptors on the bacterial cells such as proteins, lipopolysaccharide, lipoprotein, etc. And this process is reversible. The attachment process is reversible. One or more of the components of the base plate mediates irreversible binding of the phage to a bacterium. Now the irreversible binding of the phage results in the contraction of the sheath, and the hollow tail fiber is pushed through the bacterial envelope. Some phages also have enzymes which digest various components of the bacterial envelope. The nucleic acid, that is DNA, from the head passes through the hollow tail and enters the bacterial cell, and the remainder of the phage remains outside of the bacterium like a ghost. Even a non-susceptible bacteria can be artificially infected by injecting phage DNA by a process known as transfection. Now depending on life cycle, phages can either be lytic, that is virulent, or lysogenic, that is temperate. While lytic phages kill the cells they infect, temperate phages establish a persistent infection of the cell without killing it. In lytic cycle, the subsequent steps are synthesis of phage components, assembly, maturation and release. Lytic or virulent phages are phages which multiply in bacteria and kill the cell by lysis at the end of the life cycle, and soon after the DNA is injected, the phage cycle is said to be in eclipse period, during which no infectious phage particle can be found either inside or outside the bacterial cell. Eclipse phase represents the interval between the entry of the phage nucleic acid into the bacterial cell and release of mature phages from the infected cell. This phase is devoted to synthesis of phage components, that is phage proteins, and their assembly into mature bacteriophages. Now, in the lytic cycle, the phage nucleic acid takes over the host biosynthetic machinery and phage specific mRNAs are transcribed and specific proteins are made. In some cases, the early phage proteins actually degrade the host chromosome. Structural proteins which comprise the phage as well as the proteins needed for lysis of the bacterial cell are separately synthesized. Nucleic acid is then packaged inside the head and then tail is added to the head. And the mature infective phage particle is formed and the entire process is known as maturation. In lysis and release phase, the bacteria lyses, the bacteria is lysed and the new bacteriophages are released into the medium. The number of particles released per infected bacteria may be as high as 1000 and the average yield of phages per infected bacterial cell is known as the burst size. This figure displays the life cycle of lytic life cycle of bacteriophage, whereas in the lysogenic cycle there are temperate phages are involved which can either multiply via the lytic cycle or enter a dormant state in the cell. In most cases, the phage DNA actually integrates into the host chromosome and is replicated along with the host chromosome and passed on to daughter cells. This integrated state is known as prophage and the process is known as lysogeny, and the bacteria harboring these prophages are called lysogenic bacteria. Since the prophage contains genes, it can confer new properties to the bacteria, and when a cell becomes lysogenized, extra genes carried by the phage gets expressed in the bacterial cell. These genes can change the physical properties of the cell, that is the phenotype of the cell, and this particular process is known as lysogenic conversion or phage conversion. The lysogenic state of bacteria can get terminated any time when it is exposed to adverse condition, and this process is known as induction. The conditions that favor the termination of lysogenic state include desiccation, exposure to UV or ionizing radiation, exposure to mutagenic chemicals. The separated phage DNA then initiate lytic cycle which results in cell lysis and release of phages. In such phages are capable of infecting new susceptible cells and rendering them lysogenic. These lysogenized bacteria are resistant to superinfection by same or related phages, and this phenomenon is known as superinfection immunity. This particular diagram shows the difference between the lytic cycle and lysogenic cycle. The difference is here that in the lytic cycle the phage DNA is inserted into the bacteria and it overtakes the host machinery and it leads to production of new bacteriophages and the bacterial cell is lysed. Whereas in the case of lytic life cycle of bacteriophages, the DNA gets inserted into the host chromosome and it replicates along with the DNA of the host, and it is passed from one generation to next generation. Under adverse condition, this particular prophage can be excised out and it leads to formation of bacteriophage particles and ultimately lysis of the bacterial cells. Now, what exactly is the difference between lytic and lysogenic cycle is that lytic cycle leads to generalized transduction of the bacteriophage, whereas in the case of this particular lysogenic cycle, specialized transduction takes place. In the lytic cycle, random DNA of the host chromosome is packed into the head of the bacteriophage, and this random DNA gets inserted into the bacterial cell which is infected next by the bacteriophage. Whereas in case of lysogenic cycle, the DNA of the bacteriophage gets inserted into the specific region of the host chromosome, and when this specific insertion takes place, then the DNA of the bacteriophage is excised out precisely, but many a times it happens that imprecise excision takes place and DNA of the bacteriophage is taken out along with some of the genes of the host chromosome. This leads to specific transduction or specialized transduction, wherein only specific genes can be carried by the bacteriophage to the bacteria which it is going to infect next. So this is the basic difference in the two types of life cycles of the bacteriophage. Now, what exactly is significance of bacteriophages in transduction? The transduction is responsible for transfer of drug resistance, especially in staphylococci. Many species of bacteriophages, many types of bacteriophages can transfer drug resistant genes from one species to another species or from one strain to another strain of bacteria, and this leads to development of drug resistance in the bacterial species. And transduction is a very important factor in development of drug resistance, antibiotic resistance in bacteria. Secondly, lysogenic conversion is responsible for acquisition of new characteristics. Random insertion into bacterial chromosome also can induce insertional mutation. Lambda phage is a model system for study of latent infection of mammalian cells by retroviruses, and phages are used extensively in genetic engineering, where they serve as a cloning vectors. So these are some important points how the bacteriophage are used in biotechnology, or how they are used naturally for transduction to occur. Thank you. Friends, I will be talking about a very important aspect of genetic recombination in bacteria, and which is known as transduction. Earlier I have talked about how transduction was discovered as a method of genetic recombination in bacteria. What were the historical experiments which were performed for finding out what exactly transduction is and how it leads to recombination in bacteria. Now I will be talking in detail about how exactly generalized and specialized transduction occurs in bacterial species with the help of bacteriophages. So the outline for this particular lecture would be generalized transduction. What exactly is generalized transduction, what it leads to, how it happens, and secondly, what is specialized transduction, how specific genes are transferred from one bacterial cell to another bacterial cell. Then the third important aspect would be how transduction can be used as a tool for mapping genes in the bacteria. So it is transduction is a tool for gene mapping in bacteria, and how we are going to use transduction for that particular aspect. I will be discussing that, and also I will be discussing about how recombination also occurs in case of bacteriophages. So the learning objectives for this particular lecture would be that after this lecture we should be able to talk about and differentiate between the process of generalized and specialized transduction, and we should be able to do genetic mapping using transduction data, and we should have an idea of genetic recombination in bacteriophages. So these are the learning objectives for this particular lecture. So starting with generalized transduction. Transduction, as we already know, is a process of genetic recombination which is brought about by bacteriophages. In this particular mechanism of recombination, bacteriophage first infects a bacterial cell, and after that the DNA of the bacteria can get packed in the head of this particular bacteriophage. This particular bacteriophage progeny, when it infects new bacterial cells, it transfers the DNA, that particular DNA, into the new bacterial cells. This DNA which is transferred to the bacterial cells, this can be recombined with the chromosome of the host, leading to genetic recombination, which can lead to acquisition of new genes, or it can lead to formation of a partially diploid condition. How that happens, I will be talking about it in detail. So generalized transduction, as the name suggests, is basically generalized recombination. That is, any part of the donor DNA can be transferred by a bacteriophage into the recipient DNA. So this is generalized transduction. How it occurs, I will be talking about it right now. So generalized transduction is mediated by lytic phages. Now, lytic phages are those phages which, when infecting the bacteria, they lead to lysis of the bacteria. They overtake the machinery of the host bacteria, and they cause the bacteria to express the genes of the bacteriophage and make proteins of the bacteriophage. After the proteins specific for the bacteriophage has been made and a number of DNA molecules of the bacteriophage have been produced, the DNA molecules are packed inside the head of the bacteriophage, which is again made of protein, and after the head is packed with DNA, the tail proteins are attached to the head. So, we get infecting bacteriophages. All the bacteriophages which infect bacteria overtake its machinery, use it to make more bacteriophages, and in process lead to lysis or death of bacteria, and release of many bacteriophages into the medium are known as, these are known as lytic phages. So, generalized transduction is always brought about by lytic phages. In this type of transduction, any fragment of bacterial DNA can be packed in the bacteriophage head and inserted into a new bacterial cell during infection. As I have already told earlier, the generalized transducing particles completely lack DNA originating from the viral vector, containing instead only sequence of the host origin. So, the transducing particles or the transducing bacteriophages which are formed, which lead to generalized transduction, they do not contain any DNA of the virus or any DNA of the bacteriophage. They are packed only with a piece of DNA of the host chromosome. The entire DNA of the host chromosome cannot be packed into the head of the bacteriophage, because the head is a small entity and it has a limit in the size of the DNA which can be packed inside it. So, only a small part of the host DNA can be packed inside the head of the bacteriophage. These transducing particles, generalized transducing particles, arise when viral genome size fragments of donor DNA are packaged into the phage heads in place of viral DNA, and that happens when the when the bacterial cell is lysed, its DNA is cleaved into small fragments. These fragments can accidentally get packed inside the head of the bacteriophage, and when this happens, this becomes a transducing particle or a transducing bacteriophage. A random sized DNA fragment is transferred whenever generalized transduction takes place. Now, there are various bacteriophages which carry out generalized transduction in different species of bacteria. Some examples have been provided here. For example, phage P1 of E. coli is the one which brings about generalized transduction in E. coli, and it carries about 90 kilobase of DNA, which is about 2% of the chromosome of the E. coli. So, only a small fragment, that is 90 kb of the E. coli chromosome, can be packed inside the phage P1 of E. coli. Phage PBS1 of Bacillus subtilis carries about 240 kilobases of the Bacillus subtilis DNA, which again is about 10% of the genome of this particular bacteria. Another example is that of a phage which infects Salmonella. P22 is the name of this phage. P22 of Salmonella carries only 41 kilobases of the Salmonella genome, which is a bit less than 1% of the host chromosomal DNA. So, as can be seen in this particular example, only a small fragment of the host DNA can get packaged inside the head of the bacteriophages, which are going to bring about generalized transduction. Now, whatever segment is getting packaged inside the head of the bacteriophage, it may or may not get incorporated into the host chromosome. Even after the bacteriophage has injected this particular DNA into the recipient bacterial cell, there is no guarantee that this particular DNA will get integrated into the host chromosome. Stable transductants result from Rec-A dependent replacement of recipient by transduced DNA, and progeny cells will thus be haploid for the transduced region and are non-lysogenic. Stable transductants are formed only when the DNA which has been injected into the bacterial cell gets integrated into the host chromosome. If it does not get integrated into the host chromosome, transductants are not formed. Now, the generalized transduction, it can be categorized as complete or it can be categorized as abortive. As the name suggests, complete transduction, in complete transduction, the transduced DNA fragments get integrated within the recipient bacterial chromosome. So, transduction is complete only when the DNA which is injected into the bacterial cell, it is getting integrated into the chromosome of the host. This DNA fragment will get replicated along with the recipient bacterial chromosome and it is passed on to the daughter cells. That is, integrated aspect of this particular fragment within the host chromosome will be passed as such to the daughter cells generation after generation. Whereas, in case of abortive transduction, the transduced fragment does not get integrated in the bacterial chromosome and is not replicated. So, as the name suggests, the transduction is abortive because there is no incorporation of the DNA which has been injected into the bacterial cell. In abortive transduction, the DNA is passed to one of the daughter cells unilinearly, while the other daughter cell might harbor the protein product if the transduced DNA fragment was transcriptionally active in the parent cell. In abortive transduction, the DNA fragment will stay as such in the recipient bacterial cell. When this recipient bacterial cell divides, it produces two daughter cells. One of the daughter cell will be getting this particular fragment of the DNA. Now, if the DNA was transcriptionally active in the parent cell, then both the daughter cells will be receiving proteins from this particular DNA fragment and it will help both the daughter cells to survive for a few life cycles. However, it will only be transferred, this DNA fragment will only be transferred to one of the daughter cell after each division of the cell. Now, the abortively transduced DNA which is present in the cell, which is not integrated into the host chromosome, it assumes a circular supercoiled configuration within the recipient cell. That is, it is supercoiled and it assumes a circular configuration, but the ends are not sealed together, ends are not joined. So, it is not a covalently closed circle, the ends of the circle are open, though it is present in a supercoiled circular fashion. Now, a protein or proteins are responsible for holding the ends of the originally linear DNA together. The DNA fragment which was transferred into the recipient was originally linear, but now it has become supercoiled circular and the ends are held together by protein, and this particular aspect it makes it resistant to recombination as well as degradation. Usually DNA fragments which are present in the cell, they are susceptible to digestion by DNases, but because of the circular configuration, the two ends are held together, it is not subjected to recombination as well as degradation by DNases. Now, most recombination in wild type cells of E. coli and probably also in those of Salmonella typhimurium is thought to proceed via the REBCD pathway, that is recombinant BCD pathway. The recombinant BCD mediated recombination is believed to be initiated when the REC-BCD enzyme invades flush-ended double-stranded DNA and separate the strands with concomitant degradation of the 3' strand. Now, REC-BCD system is basically an assortment of proteins. This particular complex of proteins, it invades the double-stranded DNA which has been injected into the bacterial cell, and it leads to degradation of the 3' end of the both the complementary strands of the DNA. The product of REC, that is REC-BCD action, now with a single stranded tail is thought to invade the double helix to form recombinational intermediate. The REC-BCD complex of proteins, it produces single stranded ends in both the strands of the DNA, and this particular arrangement, it is responsible for invading the double helical DNA of the host chromosome or host DNA, and it leads to formation of recombinational intermediates. Finally, a primarily double-stranded section of donor DNA would replace an equivalent length of recipient DNA. So, effectively what is REC-BCD doing, that is REC-BCD complex of protein, is basically it is leading to formation of single-stranded regions on both the strands of DNA, and it is making this particular DNA attack the double-stranded DNA of the host and leading to replacement of the host DNA, a specific segment of the host DNA with the injected DNA which is coming from the transducing bacteria phage. So, effectively there is no addition of genes in this particular mechanism of transduction, only replacement of certain genes is occurring whenever this mechanism of transduction is taking place. So, rarely a partially degraded fragment of host DNA of the correct size will be packaged into the phage head and injected into a recipient. So, generalized transduction is a very rare event because the packaging of a correct size of DNA fragment which has been lysed, of DNA fragment of the bacteria which has been lysed, is a very rare occurrence. So, therefore, generalized transduction is also a very rare occurrence. The odds of a specific gene being transferred and incorporated is only about 2 per million recipient cells. So, the odds for generalized transduction are so low. First, the probability of getting of a DNA of a correct size getting packed inside the bacterial phage head is very low. Secondly, the specific genes getting transferred into the bacterial into the recipient bacterial cells are very low, as low as 2 per million recipient cells. Only if the genes are close together can they be transferred by a single phage particle. If the two genes are very close together in the host chromosome DNA, then only they can be packed together in the bacterial phage head, then only they can be transferred together into the recipient bacteria, and this also helps in mapping of these type of genes, and this particular phenomenon is termed as co-transduction. Only those genes which are situated very close to each other, they are known as co-transducing genes. The genes which are very distally situated from each other, that is the genes which are very far, which are situated very far from each other, they will not be getting co-transduced at a very high frequency. An estimate of the frequency of co-transduction can be made using this particular formula, 1 minus d divided by L whole cube, wherein d is the distance between the genes and L is the length of DNA transferred. So, if we know the distance between the genes, if we know the length of the DNA which is getting transferred, and if we are able to find out the frequency of co-transduction, we can easily calculate distance between the two genes on the DNA fragment which is getting transferred by the transducing particle. Now, this was about generalized transduction. Any DNA fragment of the host chromosome can get transferred. Any genes can get transferred. There is no specificity. Only thing specific is that only those genes which are situated close to each other, they will be getting transferred. They will be getting transferred by a transducing bacteria phage. Whereas, when we come to specialized transduction, it is totally different from what generalized transduction is. Now, specialized transduction can occur only with the temperate phages. As I told earlier that generalized transduction can occur only when the bacteria is infected by lytic phages. Specialized transduction occur only when the bacteria is infected by temperate phages. Temperate phages are those phages which can either start a lytic cycle or they can start a lysogenic cycle. Mostly they enter the host chromosome, that is, they get integrated into the chromosome of the host like an episome and create a lysogenic condition. This particular condition leads to replication of the bacteriophage DNA along with the host DNA. So, any strain of bacteria which is harboring a bacteriophage DNA in itself, in the DNA, in its own DNA or in its own chromosome, is termed as a lysogenic strain. Any phage which integrates into the chromosome of the host is termed as prophage, and all the temperate phages they can act as a prophages. The viruses which can integrate into the DNA of the bacteria, they are called temperate phages, and when integrated into the host, they protect it from attack by the same virus. Because they are already present in the host DNA, they also start producing certain proteins which protect these bacteria from attack by virus of the similar strains, and this protects the bacteria from getting lysed. In a lysogenic strain, phage reproduction is prevented by expression of a gene that represses other virus genes from being expressed. So, whenever the bacteriophage DNA is present in lysogenic condition, there is production of a protein which represses other virus genes from getting expressed, so that there is no lysis of the bacterial cell. Specialized transduction is similar to sex duction, as it depends upon a mistake made during a looping out process. Now, sex duction is a process which occurs in conjugation, wherein F plasmid which has got integrated into the bacterial chromosome, it is excised out. Now, in normal condition, F plasmid will be excised out completely as such, but in certain cases there might be faulty excision, so that along with the F plasmid some of the bacterial genes can also get excised out. So, the plasmid which is getting excised out, apart from having its own gene, it is also carrying certain genes of the bacteria. Specialized transduction is similar to that, it is akin to sex duction, because herein also there is error in the excision process and certain genes of bacteria are excised out in place of the genes of the bacteriophage DNA. Now, in sex duction, which is a type of conjugation, the error is in the F factor. In specialized transduction, the error is made in the prophage. In specialized transduction, only specific genes are recombined. Transduction of E. coli by phage lambda provides a very good example of specialized transduction. Now, in specialized transduction, the phage gets integrated into the host chromosome at a specific site. So, whenever it is getting integrated into the specific site, whenever it is getting excised out, it will only take genes from the nearby adjoining regions. Either it can take genes from the left hand side of the integration site or from the right hand side of the integration site. So, the transduction is very much specific or specialized. Lambda phage DNA integrates into the E. coli chromosome at ATT site between the gal and bio gene, galactose and biotin metabolizing gene, which are present on the E. coli chromosome. They have in between them ATT site wherein can get integrated the lambda phage. Now, if the spacing between the cuts produce a molecule between 79 and 106% of the length of the normal lambda phage DNA, then it can be packaged. That is, when the lambda phage is getting excised out from the lysogenic bacteria, and if the DNA size is 79% up to 106% of the length of a normal phage DNA, then it can get packaged into the bacteriophage head. However, if it is less than 79 or if it is more than 106% of the normal DNA length of the lambda phage, then that particular DNA fragment cannot get packaged, and these fragments will stay as such. They will not be taken from, they will not be transduced into the new recipient bacteria. Now, this figure shows how there is incorporation of the bacteriophage lambda DNA in between the gal and bio gene of the bacterial E. coli bacterial chromosome. The site which wherein the integration occurs, which is represented by light blue color, that is ATT site, and herein gets integrated the bacteriophage lambda DNA. The phage DNA can subsequently get excised from the integrated state. Sometimes the excision does not occur correctly. When an incorrect excision happens, the phage DNA carries either the gal gene or bio gene with it. The phage DNA thus formed is defective because it lacks some of its own genes. However, this DNA can be packaged into the heads because the protein products of the head and tail genes have been formed in the bacteria in which the bacteriophage was integrated earlier. So, the bacteriophage was integrated earlier in the bacterial chromosome. It was producing proteins which are responsible for forming the head and tail. Therefore, the phage DNA can get packed even if it is defective, that is, even if it is lacking a part of its own DNA and if it is carrying a part of the DNA of the bacterial chromosome, and thereby it can infect the next bacterial cell. However, after infection it cannot produce new particles of its own because it is a defective phage. It is not carrying all of the genes required for formation of infecting bacteriophage particles. Phage DNA which is deficient in some of its genes but carries some genes of the bacteria is termed as lambda D. wherein D represents defective because it is lacking some of its own gene, that is why it is known as defective. In case of bacteriophage lambda, if it carries gal gene, then it is termed as lambda D gal, that is a defective phage which is carrying gal gene of the E. coli, and if it is carrying biotin gene, then it is referred as lambda D bio, that is it is a defective phage which is carrying biotin gene of the E. coli chromosome. Now lambda D gal bacteriophage is deficient in genes coding for tail and sometimes head gene, whereas lambda D bio is deficient in INT and XIS gene. These are the genes which code for recombination enzymes. Lambda D gal transducing particles are unable to form plaques because they lack the genes of tail and head region, whereas lambda D bio transducing particles, they are able to form plaques because they are not lacking the head and tail gene. Instead, they are lacking the genes responsible for recombination process. Now, this particular figure shows how specialized transduction occurs. In a normal outlooping case, the phage DNA which has got integrated into a bacterial chromosome, it will be excised out completely. There won't be any remnants remaining in the bacterial chromosome. Also, it will not be carrying any extra gene of the bacteria. Whereas in the case of abnormal outlooping, there would be a part of bacteriophage DNA left in the E. coli chromosome, and the bacteriophage DNA which is excised out, it will be carrying a gene, one or more genes of the E. coli. So it forms a lambda D that is a defective bacteriophage DNA. The phage DNA integrates into the bacterial chromosome, and the bacteria will have an extra copy of the gene which was being carried by the phage DNA. This individual with partially diploid condition is termed as heterogeneous because it contains 2 copies of some genes. Not all the genes are present in 2 copies, only the genes which are being brought by the bacteriophage DNA, by the transducing DNA, only those genes are present in 2 copies in the bacterial chromosome, and that is why it is a partially diploid condition and termed as heterogeneous. When heterogeneous cells get infected with another lambda phage, they produce transductants which are double lysogens. If such cells are induced by UV, the prophage will be excised at a high frequency. The lysates thus produced are termed high frequency lysates. A site specific recombination event occurs for integration. As I have already told, the ATT site, it has a specific 15 nucleotide pair sequences which is responsible for integration of the bacteriophage into the bacterial chromosome. Now, this particular figure shows how a double lysogen can be produced. A lambda D gal is already present, it is already inserted or integrated into the bacterial chromosome, and a new lambda which is having all the genes, it comes and it gets integrated into the bacterial chromosome, leading to a double lysogenic condition. Such double lysogens can produce lysates at a very high frequency. Now, how do we carry out mapping with transduction? How we carry out gene mapping of bacterial chromosome with the help of transduction? Transduction can be used to establish gene order and map distance very effectively. Gene order can be established by two-factor transduction. For example, if gene A is co-transduced with gene B and B with gene C, but A is never co-transduced with C, then the order of genes will be ABC. Transduction can also be used to establish gene order and map distance, and it can also be used to map three genes on the DNA. In three-factor transduction, gene order and relative distance can be established simultaneously. It is useful when the 3 loci are very closely situated. For example, if genes A, B, C are co-transduced, the order and relative distance can be found by using rarity of multiple crossovers. For example, a prototrophic strain, that is A plus, B plus and C plus, can be used to make transducing phages which infect A minus, B minus and C minus strain. Later on, we can select only those strains which have recombined to form A plus class, that is those which have gained A plus genes. The colonies that grow in the complete medium without requirement of the A mutant are replica plated onto complete medium without the requirement of B mutant and C mutant, and in this way we can find out which particular category is least in number, and that category is the double crossover category. So we can establish that in this particular case A and C are at the extremes and B is at the center of the DNA particle. So gene distance as well as gene order can easily be made out. So this is how we done gene mapping in bacteriophages. So this is all about transduction.
Thank you so much, sir, for this informative lecture, and thank you, dear friends, for watching us. Thank you so much.